learn about plasma membrane, which is one of the topic of cell biology. In this course we going to learn about the structure of plasma membrane, how the plasma membrane is structurized and which component used in the structure of plasma membrane. So we going to know all about the structure of plasma membrane in this course. We know the nature of plasma membrane and every structural component of the plasma membrane. So what is plasma membrane? So plasma membrane is a dynamic fluid structure and forms the external boundary of cell. Why dynamic? Because it's not rigid. It's a flexible fluid structure, forms the external boundary of cell. So the plasma membrane is cover the cell externally, so it's called a dynamic fluid structural form external boundary of cell. It acts as a selectively permeable membrane and regulate the molecular trafficking across the boundary. So the plasma membrane act as a selective permeable membrane. So it's going to allow only specific molecules inside or outside to the cell. So it's a selectively membrane. It's only going to pass a selective membrane to inside or outside of the cell. So it's called selectively permeable membrane. The plasma membrane exhibits selective permeability, that is the allows some solute to cross it more easily than others. So every solute is not going to inside or outside the movement through plasma membrane because it's a selectively permeable membrane. So only the selected solutes can cross the plasma membrane. So different module proposed to explain the structure and composition of plasma membrane, but in 1972 Singers and Nicholson proposed fluid mosaic model, which is now the most accepted model. In all models, the Singers and Nicholson model fluid mosaic model, it's called fluid mosaic model, is the most accepted model of plasma membrane. In this model, membrane are viewed as fluid structure in which protein are inserted into lipid bilayer. So in Singers and Nicholson fluid mosaic model, the protein are inserted into bilayer of lipid. It describes both the mosaic arrangement of proteins embedded throughout the lipid bilayer as well as fluid movement of lipid and proteins. So in this model, Nicholson and Singers described how the protein are embedded in the bilayer of lipid, and in this fluid mosaic model how fluid pass between the lipid bilayer and through the movement of molecules by proteins inside or outside of the cell. Now let's see the structure of fluid mosaic model. So here is the structure of fluid mosaic model which given by Singers and Nicholson in 1972. So Here is the bilayer of the plasma membrane. You can see the bilayer, upper layer and lower leaflet of plasma membrane. The cytoplasm of the cell and outside extracellular fluid means outside of the cell. The plasma membrane having a protein like internal protein, peripheral protein or channel protein or pores. So, the protein which is outside or the the peripheral protein also called extrinsic protein because this protein are bound to membrane primarily to electrostatic and hydrogen bond interaction and do not interact with the hydrophobic core of the phospholipid bilayer. So, it's attached only outside of the plasma membrane. It's not going to abandon in the plasma membrane lipid bilayer. It's only a one side of the plasma membrane. So, the peripheral protein is a extrinsic protein which is bound externally lipid bilayer and it's not going to interact with the hydrophobic core of phospholipid bilayer. We learn about the structure of phospholipid bilayer later in briefly. Now let's talk about transmembrane protein. Transmembrane protein, also called integral protein or intrinsic protein, because it's embedded whole in the plasma membrane. Integral membrane protein are mostly transmembrane protein which have one or more segregate that are embedded into phospholipid bilayer. So, the transmembrane protein is embedded in the phospholipid bilayer. Transmembrane protein contain non-polar amino acid residue with the hydrophobic side chain that interact with fatty acyl group of the membrane phospholipid, thus anchoring the protein to the membrane. So, the whole protein is embedded in the lipid bilayer. It's called transmembrane protein. Transmembrane protein may be single pass or multiple pass, means it's monotopic or polytopic. So, clear, the peripheral protein is a extrinsic protein and transmembrane protein is a internal proteins or intrinsic protein. Here you can also see transmembrane glycolipid. So, what is the glycolipid? Means carbohydrate which are with lipids called glycolipid. Carbohydrates specially abundant in the plasma membrane with lipids are called glycolipid and carbohydrate bound either to protein are called glycoprotein. So, glycolipid is carbohydrate bound with lipid and glycoprotein means carbohydrate bound with proteins are called glycoproteins. Here you can see the channel of protein which are for the movement of plasma membrane a specific molecules inside or outside through the channel. It's called channel protein. In the pore, it's going only allow some specific type of molecule to enter inside the cell. That's why it's called a selective permeable membrane. So it's all about the plasma membrane structure. So what you can see in the plasma membrane structure, you see that plasma membrane is a fluid mosaic model structure given by Sangers and Nicholson in 1972, which contain a protein and lipid bilayer. In this structure, the lipid bilayer embedded throughout by proteins. The protein which are externally bound in lipid bilayer are called peripheral protein. So, peripheral protein are bound to periphery of the plasma membrane. Then transmembrane protein, which also called internal protein, which is throughout embedded to lipid bilayer. Then channel of protein, which are important for the movement of molecule throughout the plasma membrane. Some cholesterol molecule also in the plasma membrane, and then you know that glycoprotein means carbohydrate bound with the protein called glycoprotein, and carbohydrate with some lipid molecules are glycolipids. Here you can see the carbohydrate which is bound with the lipid bilayer. So it's the plasma membrane basic structural overview. All plasma membrane regardless source contain protein as well as lipid. The ratio of protein to lipid varies enormously depend on cell types. So on the cell types, it's different lipid and protein concentration. For example, the plasma membrane of human RBC contain 43% lipid and 49% protein by weight, whereas the plasma membrane of mouse liver cell contain 54% lipid and 46% lipid by weight. So, as per the cell types, the structure of plasma membrane, lipid and protein percentage are varies. So, it's all about fluid mosaic model of plasma membrane structure. The fatty acyl chain in a lipid bilayer form a fluid hydrophobic region. Internal protein float in the lipid bilayer. Both proteins and lipids are free to move laterally in the plane of bilayer, but the movement either from one phase of the bilayer to other is restricted. So it's all about plasma membrane structure overview. In the next video, we going to learn about the lipid bilayer structure. So in this you can learn the plasma membrane structure basic overview. What is plasma membrane and what its structure, having protein, lipid and carbohydrate, glycoprotein, glycolipids, etc. So in the next video we going to learn about lipid bilayer and another protein structure of plasma membrane. Enough for the overview of plasma membrane structure. Thank you so much.